All right, everybody, welcome. We are now live. I want to welcome everybody to the ECBRO Discovery News broadcast. Tonight, we have a special guest. He's on his way in now. Dr. Haskell, Hart. All right. I don't want to give too much information away. I mean, when I learned that he was gonna, he was willing to jump on tonight with us. I did some homework on him the best I could. I mean, I first learned of him through actually Richter from After Hours. He was currently on, well, I shouldn't say currently, but back in 2014, he was on After Hours with Richter and Tammy Murray. Also joined with him was Dr. Todd Disotell. I watched. It was a two-part show, part 1 and 2. First show had a lot of information. Both shows were actually very informative. I definitely recommend watching that. I actually shared that link around earlier. A lot of people not familiar with Dr. Haskell Hart. But to give you a brief rundown on who he is, he did a study. He was actually a follower of Melba Ketchum, and he tried to contact her, and after reviewing her work and actually not getting no response from her, he's actually he did his own paper. He actually has his own paper that he actually presented to her. So and it's actually broke down. I actually shared a link that he shared with me earlier. I don't know how many people got to see that or go through much of it. There's a lot of information down, and he really breaks it down. Also, we're going to talk about a few other things in general. It's going to be pretty laid back. He knows many in the Bigfoot community are not really, scientifically, I guess you could say, inclined. So he's going to, speak with us. He's going to explain who he is, what he does, and he's going to break everything down in lame terms, which should be pretty exciting. Meanwhile, while he's waiting, while he's working on getting in here, he just acknowledged he's trying to get in. So, because I have the link shared to him both through Google and Facebook. Anyway, I wanted to ask you guys. I know I've got some stuff from you earlier. I talked to you briefly. Sam, with what you and Fred were just discussing there, you've been out pretty recent with all this, with the video.
Yeah, it was today.
Okay.
Yeah.
Now you were talking about audio as well. When was the audio actually presented?
The audio, that's what the very first time I was on here, that's what we were talking about.
Right.
That video. It's more of an audio than a video.
Okay.
That's the one with the yells and the whoops.
Yeah. Now, I actually haven't gotten to listen to that yet.
Okay.
Because I've actually
I understand. Dude,
been
you're busy.
in and out.
I got it. Yep.
Yeah. Actually been out in the woods yesterday and
That's
today.
awesome. Me too. Me too.
Yeah, no,
man.
every opportunity I get, I'm out there. So
You got it.
I did
Yeah.
more or less. I did some scouting around. Me and my daughter went on a really nice hike today. So
Beautiful
yeah.
today.
Oh, it was.
Oh, dude, it was about 2 inches up on High Knob, and we got that big tree that we found that was in 2 trees, way up in 2 trees. I mean, it's an actual tree in 2 trees. I was amazed, blown away. That's in our new area up there that I was granted permission last. Yeah, but you guys need to check out. I want to do a show on that audio because it's real interesting. It all starts at the 2:38 mark.
Right.
And I think it was Sasquatch Central actually picked it up and did a shoddy piece on it, man. I'll be honest. He did a shoddy piece. He ended up taking the end of the video where my sister-in-law was in the outhouse and kind of made it look like we were fools. So most of the comments we get on there are pretty negative, talking about we're rednecks and this and that. But it all starts at the 2:38 mark, and the place that he picked it up was at the end. So guys, all of you listen to it. It's on my YouTube channel. Yeah, definitely. I know Tracy's listened to it, so.
Okay, hold on. Let me check something here. Okay, yeah. Oh yeah, Corey was just asking for the link to that information. Hold on.
Yeah, I just didn't want to interrupt him when he was talking. So, but yeah, you
Oh,
got the link
oh no,
to
you're fine.
that. Yeah, I just didn't want to interrupt him while he was talking. That's all.
Um, and for our viewers, we got viewers jumping in. Our guest, Dr. Haskell Hart, he will actually be here shortly. He's still working on getting in here. So, in the meanwhile, we're actually discussing. We're just having a little open discussion amongst ourselves at the moment. So stay tuned. Um,
Daniel,
yeah, go
Daniel,
ahead, Fred.
can you get off like maybe one day next month, first like first week and a half, first or second week of next month,
Oh,
like a
first week of.
I'm talking like a Saturday or something.
Oh yeah, any Saturday is good for me. Why?
Uh,
What's up?
I wanna ride down to his place. I know where he lives at. He lives about if you go because you gotta go down the other side of Roanoke. He lives over next to Carolina and
Right.
uh Wise County or somewhere around there.
Yeah, Wise
One
County.
way is yeah.
That's perfect.
One way is one way is 2 hours and 46 minutes, and the other way is 2 hours and 88 minutes. But I want to
Hmm.
go down. Want me to go down and check his and get him to take us to his area, show us stuff,
All right, let me check something.
and help him out.
Yeah, my video it's on YouTube. It's about 13 minutes long,
Which
and
what's your YouTube channel?
Sam Deloach. But if you go to search and type in Bigfoot vocals in Southwest Virginia, it pops right up.
Oh, okay.
Oh yeah, I
Um,
got you right here. Okay, yeah, I have you right here. Yeah.
And it starts Corey at the 2:38 mark.
Okay, it's like a 14-minute long clip.
Yeah, yeah,
Yeah,
and
it's right here.
it put earphones in, but you can distinctly hear it once you listen to the video a few times. Now, the bad thing is, a lot of people are like, "Well, why are you all talking and whatnot?" Well, for one, we're just having fun at our cabin. You know, that's what we do. We just kind of hang out at the campfire and have fun.
yeah.
And number two, it kind of freaked everybody out after it started really picking up at about the 4 minute mark. I mean, you can hear my wife say, it sounds like people are talking in the woods when there's nobody in the woods. It's our property. There's nothing for miles, and
Mm.
and you would see that whenever you come down. But I mean, the thing is, it's just something. My brother-in-law is an expert outdoorsman, and he could not explain what we were hearing. Totally could not explain it. And I mean, he's lived up there for 43 years.
Yeah. So
You
that's
know.
what. So that's why I want me and you to go down, Daniel, and get him to show us stuff, and we can teach him some simple tricks to do to get more activity. Because if you've seen that first video, he's got two perfect 17-inch tracks. I mean, perfect.
Hmm.
You could
Yeah,
see.
they're deeper. Here's the cool thing about it. Whenever I casted the print, Daniel, my father-in-law was smart enough to say, "Hey, let's make it bigger and get the ATV tracks in there."
I remember
Because
that.
yeah, if you think about a person, well, most of the time we have two people on each ATV. So think about the weight of that, and then that print's in that track, and it's a little bit deeper than the ATV track. So I
Yeah,
mean, to me, one somebody would have to be crazier than hell to come back there and actually try to hoax something on us. Number one. Number
yeah.
two, it's in like a coal pit. Is the best thing I can describe. It's like nasty black sooty coal. I mean, you just have to see it. It's weird.
But just from the first video, now when you mentioned the second one, I went to the second video, and when I seen that structure, I got two of them in my area, but they're not as high as yours.
Yeah, I couldn't believe that, man. We found that today. I mean, I was just blown away. Couldn't believe that.
It looked like the tree that that he had put up in the other tree looked like a dead locust or a dead oak.
Yeah, I mean, you can definitely see. It's
Yeah,
hard to really see from the video, but when you're there, you can see the bottom of the tree up like 40 feet up in the air. I mean, you can
yeah,
definitely see the roundness of the bottom. Like where it flares out like a tree does,
the
and
roots.
yeah, almost like it'd been ripped out of the ground or something and placed up in there.
Yeah, 'cause I got one in behind Kennedy Fields, and I got one up there behind the apple orchard.
I just want to update everybody's viewers and all. Yeah, he's still working on trying to get into the through the link. He's having some technical difficulties, but he's still trying. So I just wanted to fill everybody in because I see we got viewers. I don't want to leave them hanging if they're expecting to hear, you know, Dr.
Yeah.
Haskell Hart, you know. So
Yeah. Yeah.
I did want to ask everybody. I know I shared the link within I think yesterday or maybe actually earlier today before I went out. Has anyone gotten to watch the After Hours show? That it was actually from 2014 with Dr. Haskell Hart and Todd Disotell.
Uh-uh.
The
No, no.
link, I thought the link that I got from you, Daniel, was like 4 different subjects about it looked like about Dr. Melba Ketchum and
Yeah,
3
that's
different.
Dr. Haskell's work on Melba's
Okay.
papers and her results. But now there was another link I shared. It was actually a pod on After Hours, Richter's podcast show that he has. It actually has both Haskell and Todd on there, and they both go through and break everything down. And both gentlemen actually really threw some really great stuff out there. And Todd, I'm going to tell you what, Todd, I don't think a lot of people probably liked what he had to say, but he actually broke everything down. He kind of gave, without going into too much detail, he actually was kind of breaking down the procedures, the correct procedures, and things not to bypass in a scientific, uh, you know, study and anything when it's being presented. I mean, it's something you guys, if you guys haven't seen it, you guys got to check it out. And here he is, everybody. Welcome aboard, Haskell.
Thank you, Dan.
He finally made it in. I'm glad you enjoyed the show.
I don't do this very often, and I tried to copy the link, and it didn't copy right. Anyway,
Oh.
we're here.
Well, welcome. I was just trying to fill everybody in on, real briefly on, where I first actually, well, I learned of you through Richter, and then I actually seen your podcast on Richter's on After Hours with you and Todd Disotell.
Right.
And I was trying to explain the best I can without giving any false information on that. So I recommend them to go actually check that out if they haven't seen it. So it's part 1 and part 2. I seen both of them, and I was very impressed. I was actually blown away. I was excited, kind of like a little kid in a candy store. Like, all right, this is good information. This is what I want to hear.
The way I felt, too, this is a fairly new area for me, actually, as far as my whole career goes. I'm a physical and analytical chemist, and I took interest in this after the Ketchum paper came out, and sort of self-educated myself on a bunch of things that I had sort of heard about before, and yeah, there is a lot of excitement.
Yeah. Now, I mean, you and Todd gave some great information, and I was wondering, if you want to go ahead and start off, start off with an introduction of who you are, and then how you came about with the whole Melba Ketchum paper, and what how you actually came with your own paper, and how it was actually not acknowledged, or basically it was ignored by Melba. If you want to kind of get into that.
Yeah, well, here's the thing. I'm a lifelong naturalist in addition to being a professional chemist. I mean, I've been out in the woods looking for wildflowers and birds and trees and stuff since I was 5 years old. So I knew about the Patterson-Gimlin film, keep up with things a little bit. And then when the Ketchum paper came out, this interested me greatly. And so I decided to, since she published her DNA sequences, I thought I would check these out. And it turns out that the National Center for Biotechnology Information, which is part of the National Institutes of Health, has a database of where most people deposit their DNA data, their sequences. And it's fairly simple to use. I mean, I picked that up right away. It's well described, well documented. They have people who help you online if you're having trouble. And so I searched her sequences, first the nuclear DNA sequences that there are 3 in her paper, and I didn't get the results she got. She's saying that she got no hits, or she got a lot of different animals, and therefore it's a new species. Well, that's not true. It depends on how you do the search, and I looked into it more carefully and did some searches with some limiting parameters, and I found that this took a while because really her things matched 95% to human, and you think that's good enough, but really it isn't, and I'll explain why later. But anyway, I finally got to the conclusion that one of the samples, number 26 specifically, is actually from black bear. Number 31 is actually human or very near human. I mean, it could be a Sasquatch if Sasquatch is very near human. And then number 140 is definitely a dog. Now, just to bring people up to date, number 26 is the Justin Smacius sample.
Yeah, that's interesting.
Yeah.
I don't call it the Smacia kill because he went in 5 weeks later, 2 feet of snow with his dog, and he found this piece of flesh, which was sample 26, and there's no way anybody can prove that that's actually from what he shot. Only he knows what he shot, he and his hunting partner. And this sample may have nothing to do with it. It was found in the general vicinity, but, so I'm not so surprised that comes out to be a bear. Now, the number 140 was a blood sample on a gutter downspout, and from which there were teeth marks, and obviously the animal bled. And it's clearly a dog. I did all the work, and I think she was too quick to jump to conclusions, and I don't think she, you can say I have no biology background. Well, I'm a physical chemist. I can learn that any day and have over my life. And, she's saying I'm not qualified and this and that. Well, neither is she really. She's not a geneticist. She's not an anthropologist. She's not even a really fully trained, educated forensic scientist. She took a few weeks courses in Kentucky one summer, according to her, on one of her radio programs. And, everybody can expand their knowledge and expertise and involvement. And so I just, she called me up. I'm going to reveal this for the first time in early October of 2013. And after talking for 45 minutes about 2 inches of water in her house and how people tried to bomb her at one of her talks and lectures and all this stuff, and then finally she says, "Are you going to publish your paper?" And I said, "Well, what do you think?" And she says, I wouldn't do it because we have much more information. And I said, "Well, have you ever, in your samples, found any indication of a bear or a dog?" And she said, "Well, maybe it's Dogman." And then she laughed. She never really answered the question. And so I think she knows the truth here. Obviously, it makes her look bad, and people make mistakes. That's part of the deal. But in science, we try to correct our mistakes, and that's what I'm trying to do. That's right. The bottom line of it. So, yeah.
Now, yeah, not to interrupt or anything. Now, even Todd.
No, I'm done for now.
Oh, okay. I'm sorry. Well, I recall Dr. Todd Disotell. He made a statement about as far as scientists making mistakes, and I remember on the After Hour show, he admitted many years ago, a study he did, he made a claim off of this, but many years later with the new technology and the new, the gear, he learned that what he claimed back then was totally wrong, and he admitted to that. He was completely wrong, and
Yeah.
that's something that scientists should be willing to do, is not be afraid of being honest and saying that they made mistakes. Meanwhile,
Exactly.
you got some that I believe that just I don't know how to say this, but I guess they're maybe too proud to admit when they want to kind of follow through with what they believe what they're doing is what they're doing, but it's not accurate. Um,
Well,
now,
I don't think, if I could just jump in here,
oh,
I
go
don't
ahead.
think in Dr. Ketchum's or Melba Ketchum, I don't know if she's an actual doctor, but I think what you're dealing
Yeah,
with,
a doctor of veterinary medicine.
veteran. Okay, because I know I've seen it listed as Dr. Melba Ketchum. So
Right.
I think the thing is with her is she's not necessarily unwilling to admit that she might have made a mistake. I think with her it's more an attention seeking thing, that she figures that I can make a name for myself in this field, being Bigfoot research, and I think that's where that's her angle on a lot of things. I
Yeah.
don't know if you agree.
That's possible. Certainly could be a factor. I'm not a psychiatrist either, so I can't tell what her motivations are. And in fact, when I started this, I thought she had something too. I mean, you would think a 95% match is good, but now I said I was going to say earlier is that the way she
Yeah.
did her what we call assembly. When you do the DNA analysis, you get a lot of fragments. And, we got 3.5 billion nucleotides in the human nuclear DNA, and you get all these little fragments. They can be 100 or 200 bases long, and you got to put them together. So what did she
Mhm.
do? She used a template, a human chromosome 11 template. So you're forcing it to come as close as possible to human, and it came 95%. That means that in these DNA segments, the bear and the human have retained some of the same DNA. Not all, a small fraction actually, but there's some that's similar. Not entirely. 95% is not 100. You and I agree our DNA matches 99.5 plus each other. Okay, so 95, being a newcomer, I thought that was good. And I realized later, and I've admitted it online and everything, that that's not good enough for a species identification. So that's and she likes to quote me on my early and more enthusiastic, positive comments about her work. And after looking into it, getting educated, I've changed my mind on it. That's a simple
Mm.
fact.
Now, with what you were just talking about, referring to the DNA percentages, I know you're recalling what Todd was kind of breaking down. Now, could you basically, with all the different identical matches that fall into the percentages, there is you cannot ignore the other percentages that are within that result. Am I wording that correctly? Like, as far as like what you were just saying, it
Mm-hmm.
came up 95% human, but there's other sequences, there's other percentages, and you cannot ignore them. Correct?
Well, yeah, I think you're saying it right. It's okay, 95% human, but it's 99% bear.
Good
And the
point.
human
Okay.
that's biased, what she should have done is what's called a de novo assembly. You take all these fragments and you don't assume anything. You have to look for the overlaps. And the overlaps allow you to link two together to get a bigger one. Then you look on the ends of those and find ends of others that match and link those. You don't assume any species. And for a new species, it's just a wrong approach to assume it's it's human and go ahead and try to match it to human. Now, I've read other papers, people studying bears. Yeah, they do a bear um, reference sequence. If they're studying polar bears, they can use a black bear, and that'll be good enough to put these segments together. But when they're so different as a human and a bear or a dog, it's just the wrong approach, period. And other people who are experts in the field have said so.
Okay. Yeah, go ahead. Fred has
Is
a
that
question
making
for you.
sense?
Oh, yeah. I understand very clear. But go ahead,
Good.
Fred.
Great.
Yeah, hi. My name is Fred Caney. I run a Bigfoot group down here in Virginia called Valley Bigfooting.
Oh, yeah.
Maybe you can answer this question. In '81, I had a Squatch come out 5 feet in front of me.
Oh.
His face looked more human than it did anything. I mean, to be the size he was, looked like a brick wall. But his nose, the bridge of his nose and his nose was not flat, but it was, kind of like a small nose but wide, and he had thin lips and everything. But what I noticed is, I looked at him from his head, his eyeballs, all the way down to his feet, and everything looked like a human on that thing. So, am I wrong in saying that it could be 40% human hybrid and the rest of it be a hominin or something like that?
Right. Well, there are a number of possibilities. It could be if there's such a thing as Bigfoot, and a lot of people like you have seen it close up. It could be that it is a very near to human, either hybrid or other hominin, and it could also be a human with some genetic defects.
What is the word? I can't remember. Scott Carpenter mentioned it's called Kukakis or something like that.
Okay, for a hairy individual, trichosis.
No, he said it was human hybrid mixed with Kukakis. That's it, Kukakis.
Or cognatus.
Oh, cognatus.
Yeah.
Yeah.
Well, cognatus is the word that Ketchum gave to the new species. She called it human Homo sapiens cognatus.
That's it. Okay.
Yeah.
Okay. Now
and that's just her word for it. That doesn't mean anything, and it hasn't been accepted officially anyway. So, but this condition that certain humans have, it's very rare, where they grow heavy hair all over their body. It's called trichosis. Okay?
Yeah. Now with this one here, he had hair on the under part of his arm, probably about 6 to 8 inches long, and from his shoulders up, looked like he took a brush and teased his hair. But his hair on his upper arms were thin, and he had dark skin, like not quite a black person, say like an Indian who lives in the desert, like Apache Indian. How dark their skin is. That's what he looked like. That's what the color of his skin was, and everything.
Yeah. Well, that's not too different from what some other people say they've seen too. So, what it exactly is, I don't know. I don't think anybody does for sure. What we need really are samples that can be taken with some video or some pictures to go with them, because you go out in the woods and you collect hair samples. Well, they can be a number of different kinds of mammals, and you don't know if they've been contaminated either. Now, Melba Ketchum is very careful to say all her laboratory procedures eliminated contamination. Well, that's fine in the laboratory. But she didn't know what happened before they got there.
Yeah.
Yeah, because
whatever.
Yeah, because I know Daniel's gotten samples out of my area, and I've gotten samples, and I know we've sent them. I know we did the proper thing to send them off. And I've sent mine all around. I've sent 4 samples off in the United States and Canada, and I have yet to hear from them. This is in the past 2 years.
Where did you send them?
Oh, gee, I can't remember half of them now. One, let's see. I know there was two in Washington State, and one in Oregon, and one in Canada.
Okay. Were
Now,
these scientists you sent them to?
I
Or,
know some guys I met online said send them to these these colleges or
okay.
institutes.
University, yeah.
now,
Or, go
now,
ahead, Daniel.
my samples, the first set of hair samples I first collected, which was what was this, about 2 years ago, Fred, out of
Yeah.
your area.
Yeah.
Well, yeah, I've collected some samples, and you know, and I got in contact with Dr. Jeff Meldrum, and I want to know. This is when he was accepting, some samples, and well, he instructed me on the proper way to, send them in the envelope, and so I did, and of course, I knew I wasn't expecting to hear nothing anytime soon. Within, but months were going by, months and months and months, and then here came a year, and then I was like, I did try to message him, try to say, hey, have you heard, is there any results on that? I was curious. Now, when a local newspaper group, when they were doing a study, oh, not a study, excuse me, when they were doing a report and story on my group, the ECBRO, well, when I shared some information with a news reporter that, what I've done and, the samples I've collected, she apparently was able to get in contact with Dr. Jeff Meldrum, and he actually kind of shared information with her on my results. And my results actually were black bear hair, which no problem. I was glad that, long as I knew the truth of what they were, I wasn't going to argue with what they were. I mean, yeah, I would have loved them to be something different, but, but, I didn't hear directly from him. I had to learn that through the newspaper reporter who did the story on my group. So, but,
Yeah.
One of the hair samples I did. I remember. I did send one sample to Jeff Meldrum. You know how when you take and pull a hair, you got that little root at the bottom
Yep.
of it.
Right.
I got in touch with him, and he told me to send it to him, so I sent it to him, and I have yet to hear from him.
Well, I'll tell you what. I think it's really bad treatment of people to not inform them about the progress. If you can't do the analysis, just tell them. And certainly, if you've done the analysis, they should be the first ones to know, the people who gave you the sample. And so that doesn't impress me too much. I'm sorry about that. Now, I will tell you that Meldrum contacted me recently in the last month or two and asked if I had any recommendations for where to send samples for analysis of this sort, DNA analysis. He doesn't do DNA
right.
analysis in his lab, And I said, well, I recommended it to him to get Washington University in St. Louis, who Melba claims is reanalyzing her samples. I don't necessarily believe that, but whatever. They have a good facility there. I don't know what they charge or anything, but I did recommend that. And there aren't that many people that are willing to take something like this on because they can get thousands of samples from people, and who's to know which ones are the most likely ones to produce good results. So I'm not surprised you didn't get much, but they should just tell you, we can't do this, our backlog's too heavy, we need funding to do it, can you raise some money, or we'll send it here and let them deal with it if you're willing to pay or whatever, but I think it's bad to not keep you informed on it, and certainly to give results to somebody else before they give them to you. I don't think that's ethical.
Well, and that's
Right.
kind of one of the things with the DNA testing is sending hairs in. It doesn't cost $20 to do it. It's like a few $1,000s, isn't it?
Right. Typically, if you do the hair, you're going to do the mitochondrial DNA. And, Melba used a lab that I've used too. It's called Family DNA. You can get an analysis. It's not a complete DNA analysis for a couple $100. And for a little bit more, you can get the complete mitochondrial 15,000 16,568 bases, the whole thing. And that's several $100s, 3 or something like that. But they're a reputable outfit. But you know, they did her work, and there were some flags. Like, there is such a thing as it's called the philo tree. It's a tree of life of the mitochondrial DNA of all humans, and they only differ by a couple tenths of a percent. Okay, but this shows you all the different mutations, and typically you have to be somewhere near one of these branches within typically 4 or 7 bases out of 16,000 of one of these tips, or somewhere in there. And some of her samples were 16 bases off. They're really unheard of. They're human-like, but they don't match the chart. And so that should have been a flag. I think it's due to contamination species, and so the analysis didn't go quite right. I don't know for sure, but they should have flagged that. She put out a table, supplemental table too, with all those results. I looked at them in great detail, and there's some things there that don't look too good. They just they can't be human, but yet they're closer to human than anything else, you know. So I don't know. Some more work, definitely some more work.
Yeah.
I'll go ahead, Fred. Go ahead, Fred.
Yeah, because one sample I took to the we got a place down here called the Wildlife Center. It takes wounded animals and gets them healthy and releases them back in the wild.
Yeah.
And I took one sample to him, and I didn't hear from him, but Bill Brock was in my area back a few years ago when he started that show about when he goes into caves and stuff, mysteries or something like that. And he asked about DNA from around the area, and he gave him my DNA, and it said human contaminated on it or something like that. That's what he told Bill. Said
Yeah.
He said it was like someone touched it.
That's funny. That brings me up to my next question, Haskell, if that's okay to call you.
Sure, please do.
Okay.
Yeah.
Well, I think a lot of people when they hear contamination, especially in this whole Melba Ketchum situation, human contamination. Now, maybe I might be under the wrong impression or the understanding of it, but contamination is exactly what it means. It's contaminated. Now, is this correct? Or, like, for example, if I give off, if I collect the hair samples and did not do it properly, if they were to actually test it and get human, well, there's a difference because there's a bacteria that causes the contamination on it. Now, could you kind of give us, fill us in on actually contamination, and is contamination considered DNA? Which I like to believe it is in a way, but
Right. When they talk about that, they mean it's contaminated with some other DNA. Okay? So you have a hair, maybe let's say it's a bear hair. But in the process of collecting it, and you don't know, it's been sitting out in the woods, a human could have gone by and sneezed on it,
Mm-hmm.
And then you go and analyze it as if it's a human, and you're going to get the results of that human being that contaminated it. And you're going to get all excited and you say, I found this human-like piece of hair, and Sasquatch must be a human hybrid and this and that, when really you're looking at the wrong part of the sample because of the way you did the analysis, and you assumed it was human, and that's what they have to do. And so you've got the human contamination part of it. And if you backed up a little bit and did it with a more open mind, the analysis, you'd see that there was more bear there than human. But you're right, the contamination is contaminated DNA.
Very good. I'm glad. I like the way you broke that down. I like to feel it's safe that hopefully everyone understood that.
I did. I understood.
Because that's something I've been trying to get across. I'm not no scientist by no means, and I'm not I do understand a few basic things, and you know, and that's one part of what I've been trying to get at for a long time is, and talking, you know, as far as I've said it before, Melba's cult. She has a lot of followers, and a
Yep.
lot of them. I've been jumped on a few of them, you know, by a few of them, and one in particular. But you know, I don't try to attack people when I present stuff, and you know, because I started off asking, questioning this whole thing because it did not add up in my book, you know, and so I started throwing things out there and started presenting things, and then people try to throw things at me and, well, you know, try to use her own study and everything. People don't understand. Her followers are, some of them think they're scientifically inclined and they're not. And I've had people, do you understand the paper? Do you understand science? But you know, have you even seen her paper? I've looked through it briefly. I don't. I saw too much that I didn't even want to bother looking at it no more. I mean,
Yeah.
you know, like I said, I'm not a scientist, but I do understand some terms and, you know, whatever. But so I think a lot of people are kind of mixed up in this whole thing and understanding that they're going by hearsay more than anything. It's because she's saying it, they're believing it. You know.
Yeah.
I do have a question about the contamination thing, if I might. How
Yeah.
how common would how common is contamination in any sort of, say we're talking about hair samples, like if I have a cat, I grab some hair off of its tail and I send it off to be analyzed. Now, would that, if that came back as cat with human contamination, could someone, not myself, but if someone was into that such a sort of thing, could then turn around and say it's some sort of odd cat human hybrid? And is that what we're dealing with in the whole Melba Ketchum thing? Is that it's contaminated, but she's focusing more on the contamination than the actual DNA of the hair sample itself?
Well, that's part of it. Yes. So here's the thing. The most common type of contamination is from bacteria. And you know, any kind of animal, including us, is going to be full of bacteria, and that has to be considered. And there are ways to clean samples. And like for a piece of hair, you can wash off the outside and then extract the DNA from the center, from the medulla of the hair, and you know, do pretty well. But it is not easy. People who've done this for years with ancient samples, especially, are difficult, that have been in the ground for thousands of years, and so on. They have certain very rigid procedures they go through, and it's not easy. It's not something somebody can just walk into and do in a laboratory for the first time. It takes a lot of preparation, the right types of. They even have positive airflow out of these laboratories so that air doesn't come in; it always flows out, and so that you don't get things in. It can be airborne contamination. You
Mm.
know, somebody sneezes, all that stuff is full of bacteria and your own DNA and everything. So yeah, it's a problem, and I don't think she took it seriously enough, especially for a first timer on something like this. Working with other people who hadn't really done this sort of unknown identification before. A lot of her authors are, they're criminologists. They know they're dealing with human. They look at human DNA at a crime scene, you know, and that limits the possibilities and makes it easier. But
Mm.
you can't use the same procedures on an unknown.
Right. Now, that actually will bring me to another question. But before I ask my question, I'm sorry I've been holding off on Sam Deloach over here. Sam, just got a question he wants to throw out.
Okay.
Hey, Dr. Haskell. My name is Sam Deloach. I'm a researcher in High Knob, Virginia. It's a pleasure to talk to you.
Pleasure likewise here.
Hey, I just a little bit confused when about the when you said that she tested these samples against a human template. What template or templates do you usually test them against if it's not a human template that you would use?
Okay, let me try to explain that again. If you know pretty much the species you're dealing with and you're just looking for some mutations in it, or some hybridization. When you know it's a bear or whatever, you can use, say, a bear template, or you can use a human. If we're studying human diseases and we want to look for slight changes in the DNA, we can use a human template because all our samples we know are human. We took them from blood from somebody's arm. But when you don't know the species, you use a different, you don't use a template because that's going to bias it. That's what she did. She got 95%, a bear that equaled a human 95% because she used a human template. And only those parts of the bear that are like human, because it is a mammal, it's got some similar function, only those parts were sequenced. And so the appropriate approach, you can look it up, and I don't want to get into it too much. I'm not really an expert in it, but it's called a de novo assembly, and it doesn't assume anything. It looks for overlaps in these segments, okay, and puts them together that way. Let me give you a simple example. If you have only two, and one is ABC, and the other one is CDE, they have that common C. So you can put those together and say you have a sequence of ABCDE because of the common overlap of C. That's extremely simplified, but that's what they do, and they don't assume that it's from any animal. And that's what should have been done, and she'd have seen right away that it wasn't a human or anything like a Sasquatch human hybrid, that it would have been a bear or a dog or whatever I found there, depending on the sample. So you see what I mean? If you force it to be human, you're going to get those parts of the DNA that are similar to human. And because we're all mammals, we all breathe, we all have certain muscle functions and similarities, there's going to be some DNA that's similar. Not exactly, 95%. And that's where at first I got deluded into thinking that this was great and she had something, till I realized that's not good enough for a human match. You got to be 99.5 plus.
I personally understand clearly what the way you broke it down earlier, and I really appreciate that. Now, one of the other questions, one thing I wanted to state before I ask my other question, when we were talking about cognatus, whatever, what Fred was asking earlier, I remember, if I remember correctly, Todd Disseltel actually spoke up on or brought up the Zana story, how everyone thought she was not human. She had Neanderthal in her, and after doing some testing, it came back as not but strictly all human. She was African descent.
Correct.
That had that disease, that whatever, I don't know if it's a disease. I don't know if that's the right way to say it, but she had that, whatever you want to call it, that caused her to be all hairy, right? So
that's correct, and that work was redone recently by Brian Sykes, professor of genetics at Oxford, and he came with a little more detail. He had better equipment available than Todd did at the time, and that's still correct. Now, as far as the hair, I've read stories where kids get lost. There were some cases in Africa, and the monkeys raise them, they give them food, and when they come back, they got hair on their body. So it can be an adaptation to protection from the environment, especially in cold weather. And it's amazing to me that a human could grow long hair just by being out in the woods long enough.
So it's almost like a type of adaptation, the way they adapt to it.
Almost like a feral adaptation.
Yes, exactly. You're a feral human if you're out there for any length of time. And that kind of confused people into thinking, that she was some other species. But it's a very interesting piece of work. They found relatives, grandchildren, and great-grandchildren, and they compared the DNA. They found the skeletal remains of her son, which they extracted DNA from the bones. And it's all a very tight case. And how an African haplogroup would get there into central the Caucasus in Russia is interesting. One theory is she was an escaped slave from the Ottoman Empire, which controlled that region for many years. Or the other one is she could actually be one of the descendants of the people who came out of Africa that we're all related to.
And for the record, as we, as Corey brought up, possible feral. For those, for viewers or anyone who will view this in the future, I've heard the word feral being mentioned up in possible theories of, we've heard of stories of feral human from here and there. Feral humans, possibly, for those who might believe in that and along the Sasquatch theory, well.
Yeah.
Feral, I mean, I think it's safe to say that feral is not a gene. It's not a DNA. Is that correct to say that?
That's absolutely right. That's a mode of existence, and it is not a DNA characteristic. So a feral human is going to have human DNA. It's going to be just the same as it was before he left his environment and went out into the woods, So that's correct. It will not show. You won't see a difference.
A question I've always wondered about is that if you took DNA, a hair sample, because we've been talking about hair samples, if you took a hair sample from me and tested it, and a hair sample from, say, a mountain gorilla, a close relative, and you did DNA testing on both, what would be the differences that would show as far as my hair sample and a mountain gorilla's hair sample? Would you be able to look at them and go, this is a mountain gorilla, this is Corey, basically?
Yes. They would differ by 5 to 10% only. And you would be able to tell because other people have done the same thing and have deposited their data in the GenBank database, and you could compare either sample to that database, and it would tell you that yours is human and his was gorilla. Okay? There are definite differences, and they're well known. They're in the database. They've been well studied, and the differences would be in the 5 to 10% range.
Oh, okay, interesting. There was something I always wondered about that. If we're so closely related, how would you be able to tell between, just from a DNA sample?
Yeah, the DNA sequencing is very precise. There are the 4 bases. They have the symbols A, G, T, and C, and it's their sequence that matters. And 5 to 10% is definitely a large difference in this game.
Okay. Now, if I've seen, I hate to bring up stuff I see on television, but of course they have, as far as genealogy goes, they have some services now where apparently you can submit, I believe it is a hair sample, and they could test me and say, okay, he has European lineage.
Right.
On one side of the family is from Poland. Do you know if anything's been done with some of these apparent Bigfoot hair samples as far as that goes?
Yeah, well, usually for you or me, if I wanted to check my genealogy out, we'd take what they call a buccal sample from your cheek.
A swab inside the mouth, okay.
Because there's more DNA available, and it's a little difficult from a hair. But yes, they can take hairs and do, the preparation is different, but the sequencing and the methodology after that is virtually the same, as I understand it. So yeah, you can use hairs, but for practical reasons, for people who have a lot of sample, they take it from your cheek. And these are reputable companies, and what they do is first rate. I've looked in the one I'm most familiar with is Family Tree DNA, and yeah, you can. People get surprised. They think they're Italian, and they got 16% Italian, but 84% something else, you know?
Yeah, I've seen a commercial on TV where the guy was always like, "Oh, we always did everything like we were
That's
German,"
right.
and
That's right.
and then I did my testing, and it turns out I'm Scottish and Irish. Because
Yeah.
the first thing I thought, being a Bigfoot hobbyist, is like, "Geez, I wonder what would happen if you sent in a hair sample to these people."
Yeah.
They don't do like you said. It's mouth swab. I guess is a little more of a easier
Easier for them to
material
deal
to work
with.
with.
Yeah.
Okay.
Preparation is easier, yeah. And they put it in a solution that preserves it, and you send it. I've done it. I have mine, and I have other people in my family, and a few, and so on. And so that's the way that goes. And I'll tell you something. I submitted. They would hate me if they knew this. I submitted a dog, a cat, and a horse sample to this family tree DNA. And to their credit, they could not sequence it.
Really?
They said you need to give me another sample. And
Oh, that's good. At least they stepped
that
up
is
and
good.
said, "Yeah, this isn't right." Well, that's good. That's good.
Yeah.
Good to know.
If it's not human, they won't be able to give you an answer. Otherwise, it would be a little more confusing. And so, and that means too that it wasn't contaminated greatly by human. You know, I got them from friends of mine with their pets, and we did it carefully as we could. But there was not enough human DNA in there to sequence because their methods are geared toward human, okay? And now, if you were to take that result and say we got nothing, well, somebody would say, oh, it's a new species. You know, they couldn't get anything, it's a new species. No, it's a different species that their method just doesn't sequence.
Hmm.
It's only for human.
Now, I had a question in regards to Melba's co-authors, um, or I guess trying to understand this or actually word this right at the same time. On her peer review, on the peer review, she has what you call co-authors who sign off on the study. Is that correct?
Well, there's a difference between peer review and co-author. Co-authors are authors on the paper, just like the primary author. She's the primary principal investigator, and they're also on the paper, okay?
All right.
Whereas peer review is by people you don't know, who are also professionals in that area, and they're selected by journal editors for their knowledge of the field, and they provide their comments anonymously, and then the author or the authors have to consider those comments and revise their paper if necessary, or give very good reasons why they're not, and that means something that's very good, that means that the people didn't understand what they said or something. Usually they revise. So peer review is by anonymous third parties who don't, they know who you are, but you don't know who they are as an author.
All right. All right. Well, that will bring me to another question here. Now, well, first of all right, on the peer review, um, all right, I'm trying to think of the best way to ask this.
Yeah.
Well, maybe I should ask with the authors. Now, the authors, they're all right. They're responsible for the work she's done just as much since they co-signed. I mean, is that correct? That they're responsible for what she put out?
That's exactly correct. You're, as all the co-authors have to agree with the results and with the written paper.
Okay.
And while different ones may have done different things, and maybe expertise in parts of it, they're all totally responsible for the whole paper, and they should be very careful about agreeing to be a co-author without understanding the whole parts of the paper. You know?
Now, could this affect them by them being co-authors by signing on her work? Yeah, like, I mean, how would this affect them as far as if the results are not what she's claiming they are? As far as she's being pretty. I mean, what she's putting out there is a pretty blunt and bold claim. But you broke it down and explained how it's she didn't do everything completely right. I mean, she did things out of error and not looked into the whole sequence correctly. So, I mean, how does this affect the co-authors, or does it?
Well, they've been extremely quiet about it. I've tried to contact them. I've sent them everything I sent her with the work that I had done early on, and I got no responses. I think, they're hunkering down, is my opinion, and hoping it'll blow over.
Right.
Silence can
Only
speak.
one gave me I asked the microscopist why he thought that his pictures showed some double-stranded DNA, which is normal, and some single-stranded DNA connecting the double-stranded. And he says, I don't make comments about cryptozoology on my microscope, which I think is very poor scientific ethics and attitude. You submitted those photographs, your name's on the paper. You ought to be able to tell me something about what you saw. You don't just throw stuff over the fence. And I said, could it be microbial degradation? Very likely. Viral attack, gene splicing, to go way out, or something else. And he wouldn't even give me any ideas. He's just a microscopist, and that's the problem with these authors. None of them are expert in what this paper is about.
Mm.
Those little parts that they had involvement with, but that's it. So they don't see the big picture, I'm afraid. That's my
Right.
view.
Well, that's very good. That's very interesting as well. So now, all right, now with the peer review, all right, because I'm asking so I can get a better understanding, as far as viewers as well, now you explain what the peer review was. Now the peer review, well, you might have to kind of break it down and explain again. Now, with the peer
Okay.
review, if that's something that's submitted, is that what gets approved?
Yeah, you submit a paper to the editor, okay, of the journal, and he looks it over. He may consult with some co-editors, and if they think it's generally something in the area of that journal and looks at least likely to be reasonably accurate, they will send it to usually 2 to 4 peer reviewers. These are people in the same field who would be familiar enough with the subject to be able to make critical judgments about the work. Is it accurate? Did they use the right procedures? Did they draw the right conclusions from the results? And is it readable? And are the figures readable? And could a person with maybe a little bit less knowledge of the field read it and understand it? And they
Okay.
submit their comments. They're very precise points and referencing very specific things. They sometimes make general comments too, but really the guts of it are the critical point by point. You said this, but then you said that sort of thing. Or
Right.
this does not imply that because it could also be this. Or this figure isn't clear. You forgot to put the units on it. Or you forgot to say, give it a title. Or you need to put some footnotes on the figure because it's not clear. Or that sort of thing. Or this table is too big. You need to compress it or submit it as a supplementary additional data table. That sort of stuff.
Okay.
And
Now,
then the authors respond, and the peer reviewers look at the response, and they either agree with the response and the changes, or they say, no, you still have to do this, this, and that. And this could go back and forth a few times. Hopefully not too many.
now, are you still? I mean, as far as currently, are you still trying to follow her current work? I mean, like as of now, are you aware of anything that might have changed with her, or basically everything's still the same as far as what she's trying to do?
Yeah.
Yeah. As far as I'm sorry. Go ahead.
I'm trying to keep up with it.
Right.
Most of what she's done since the publication in her de novo journal has been interviews on radios. I would call it sensationalist radio. People who they like to do far out topics, and that's okay. I don't have any issue with that. The only problem I have is they're taking everything she says at face value. They're not asking for any other comments, any criticisms, although they know that they're out there. And it feeds their program to have this sensationalist stuff. And same with some of her TV interviews, although a little less so. But she hasn't published anything else. No retractions. I follow her Facebook. I follow, whatever news I can on it. And I will say now that I have put together a compilation of all my work, and it's been accepted in a peer-reviewed journal. I'm not going to give details because I don't want her running out there and trying to sue them and sending letters and trying to sabotage it. When it comes out, I'll send her a copy. So in the spring of this year, it's supposed to be published, and I'll let everybody know. It will be available to the public, and I'll have a few extra copies that special people like you, Dan, will get a free copy of.
Very good, awesome. That is awesome. Now got to oh man, where was I going? I got excited there for a second. Lost my train of thought.
Peer review and following her work.
Yeah. So okay. Now.
Well, the thing is with the most recent things I've seen with Dr. Ketchum are GoFundMe pages for further research on the DNA testing.
Yeah.
What else can you do? I don't.
Okay. Well, she claims to have some more samples, and she claims to have some samples from giants. Various unusual large skeletons, some of which have been reported and none of which seem to be available in any museum that I know of, but she claims to have some samples. And also she's interested in this Paraca, I think it's called, skulls in Peru, the elongated skulls.
Yeah.
Oh, yeah, okay.
Doing some analyses on that, as is Brian Forster, who's also well, I'd say he's an amateur archaeologist, but that's okay. So she's trying to analyze these new samples.
Now, I'm sorry. Go ahead. I didn't mean to interrupt.
No problem. Go ahead.
Now, what I was going to say is now, as far as what you just stated, as far as she's claiming to have DNA or whatever, some samples from, some giant skeletons. Well, I mean, I believe it's safe to say, now, I believe in the Bible and a lot of, there were giants that roamed the earth at one time. I mean, we still have tall people. I mean, and that's all they were, was no more than people. Now, the elongated skulls, I mean, in some countries or, in burial, I believe it was I don't know if it was a tradition or whatever, how you want to call it, but a lot of people had the skulls wrapped to be formed in a certain way.
Deformed, yeah.
Right, exactly. So I mean,
some people say because of the size, shape, and certain other anatomical features of the skull, that they could not have been formed by deformation of a normal skull. I don't know enough anatomy to be able to make comments on that, but there is a view that some of them could not have been normal deformations.
Interesting.
I don't know the truth of it, but
Well, you have the whole what is it, the Japanese foot wrapping?
Right, the Chinese, yeah.
Where Japanese women back a few hundred years ago, they would wrap their feet so the toes were all tucked in, but I know Ketchum seems to have gotten into the whole Nephilism thing.
Nephilim,
and that, then he just seems to go from one anomalous thing to the next. So I have a hard time taking her seriously, personally, myself.
Well, she's not getting very much money as this kind of thing goes. If you look at her Facebook page and all her
Yeah.
appeals every couple days for more money. She's also raising money for a movie she's going to produce called Bigfoot Decoded,
Hmm.
which I'm assuming she's going to explain all the DNA work that she's done and try to justify it to the public. And that needs funding too. She's trying to raise money for that. And we'll see what she comes up with.
I seriously doubt if she's going to have too many people with counterpoints to her theories and whatever she's doing. So,
Yeah,
unfortunately, I was actually excited at first when I heard, and then I started reading more into her stuff that she was doing, and I just got disappointed again, as you often do in the Bigfoot
right.
community.
Well, my experience is very similar. I was excited too because I mean I'm a naturalist too as a hobby, and to have this huge new species, uh, a hominid at the least, and it was exciting. I thought, well, gosh, I need to look into this and see if I can agree with her conclusions, and that's how I got involved really. And I've learned a lot about it. It's still a lot to learn about it, but yeah, I'm sorry. She says I fell in with the haters. Well, I don't
Yeah.
hate her. I mean, she's not. I hate people like Stalin and Hitler, and you know,
Yeah.
she's not important enough to hate. And I
No,
certainly,
disagreement. That's a whole internet thing. The haters, the haters, the haters. I don't hate her either. I just disagree, and it's
yeah,
just come now that if you disagree, you're a hater. I don't hate her at all, but
yeah,
it's just obvious what
I
she's
agree.
doing. She's trying to make a name for herself in this small community, and she
Right.
won't let anyone take that away from her.
She's trying to marginalize you and make you look bad just because you don't agree. You're exactly right. And no,
It's
that's
it's
very
unfortunate
unscientific.
because that's why a lot of people involved in Sasquatch research aren't taken seriously,
Yeah,
because that's who they're going to point to. They're going to point to Dr. Ketchum. They're not going to point to someone like yourself, because you bring a little reality and common sense into it where some folks don't, unfortunately.
well,
Well,
it's
it,
not a very well. What should I say? It's not always a very comfortable position to be criticizing somebody else who did some original work. It would be much better to do your own original work. I don't have a laboratory for this kind of research, and all I've done is computer searches, really, that anybody could do. It's free on the internet, and I've done the most I could with the data that she presented. It's her data, her raw data, and I don't fall in with anybody. People who know me say would agree that I'm a very independent person, and I'm not likely to be swayed by a bunch of people. I may agree with them, but I'm going to have to come to my own conclusions, as I think you guys are too, from what it sounds like, what I've heard today.
Yeah, and that's how I've always approached it. Show me what you got, tell me what you got, I'll give you my opinion. If I disagree, that doesn't mean I again, the whole hater thing. It doesn't mean I hate you. We just disagree.
Yeah,
right.
right.
You know, a lot of people, like I mentioned earlier, when you question science, you know, the, I don't know if this is the right way to say it. Um, when you question science, I think the uneducated kind of, I guess you could say, they're the ones that start throwing back negative comments because they're not properly trained or educated in that field, and they think you're the one that's not educated in that field. It's not that you're questioning things. I believe in science. Science should be questioned. And
Absolutely.
and all science is not not correct anyway. There's theories that float around. I mean, as far as I'm concerned, there's theories that a lot of people like to push on people, but they're not proven, and that's all theories are. It's
Yeah.
theories just like a hypothesis. It's an educated guess, more or less. But
Right, and science continually evolves. I mean, this is not my idea. Everybody knows that. And so some things are going to be proven to be not correct, or they could be proven to be part of a more general, a better theory that explains more different phenomena. And you just have to, if you can't accept that, then you don't belong in the business. And so, you know, um, that's the way I see it. I mean, I don't take her personal attacks on me seriously. I mean, they're the signs of a desperate woman, you know, trying to reclaim credibility, and so that's fine. She can criticize me. I think my credentials to do what I did are better than her credentials to do what she attempted to do.
Right.
All I did, if you break it down, is compare a sequence of 4 letters. I mean, it sounds simple, but there's a database and software to do that. But you have to use it intelligently, okay? I didn't do any lab experiments, and I made that clear. I don't have that capability. So that's where I stand on it.
Now, recalling back, watching the show you were on with Richter and Todd Disotell. I mean, I've watched and listened to both of you guys. Both of you guys were in agreement with each other, and Todd broke it down, and even though he was speaking scientifically, I think he said it very clear. I mean, I don't think he could have said it any clearer than how he put it. And, and I think more and more people need to see that. I know I shared that show link around earlier, and,
yeah.
And of course, I don't, for our viewers watching, I hope they understand what you threw out there and how you're putting it down. Because more and more people need to hear this. They need to understand the procedures and what wasn't done and what was overlooked, and the real results, and I think, a lot of people just, don't. I think you're still going to have people, even if they do understand what you said, and even if they listen to Todd as well, you're going to have people that might understand, but they're going to deny it, and they want to keep following because they want to believe what Mel was saying. So,
right. Well, there's believers, and they're not, a lot of them, ever going to change their mind. And because they saw something, that's fine. I'm not denying anything that people said they saw, because I wasn't there, and that does not mean that this particular individual study was done correctly and came with good conclusions from good results.
Right.
Maybe there is a Bigfoot out there,
I mean, there's some of us that, I will state, I'm not just a believer, I'm a knower. I mean, and, I know a couple others. We've actually, Fred's had some experiences, and there's many that claim, and actually, some of them are very trusted, well individuals, real good, other good, close friends of mine, who actually had one about 2 or 3 months ago for the very first time out of all his youth. He wasn't expecting to see anything like that, but he was, he's been on Night Callers Radio. We've had him on our show here, and I believe without doubt he saw what he said he saw. But I mean, it's just the creature is a very intelligent creature, I believe.
Well, I respect these observations from thousands of people, really. And you have to think there must be something to it, because they can't all be. I mean, some of them could be mistaken, but there's enough of them that I have to believe there's something to it. And I personally think the Patterson-Gimlin film is authentic. I've studied in detail the work of Bill Munns, who wrote a book on it. He's a costume expert from Hollywood, spent his whole career on costumes, and he says that the muscle movements and all kinds of anatomical features could not have been reproduced with the costume materials of that day.
Thank you.
Have spandex, okay?
Thank you.
And I have to believe him, because he's an expert in that. Until somebody goes and point by point refutes what he said, I'm going to believe it. And because it makes sense. It may not be correct, but somebody's got to do a whole lot better job than these people that just say he's putting a bunch of nude women across the stage and making conclusions about their bodies and stuff. But it's anatomy, and I don't know any other way to do it. But so maybe that'll be proven to be false someday, but I don't see it at the moment.
Right. I mean,
I mean,
I,
Well,
they've done,
to
they've
me,
done,
kind
they've
of
done.
show that it kind of depends who you talk to. Like I emailed a couple of different special effects people in Hollywood. Tom Savini, who was Creep Show, Friday the 13th. He's done a bunch of movies.
Yeah.
Everett Burrell from K&B Effects, and I sent two or three people, an email. Did you hear that such and such a person created this suit? Would it have been possible to make a suit like that back in '67? Could you make a suit that looks exactly like Patty to this day? And the three people that I emailed all said yes. So
That you could have made it in '67 or could do it today.
with the technology available in '67, could you go back and make a suit as convincing as Patty? And all
No.
three of them said yes. One of them even said yes, it is possible. That suit is fake. Philip Morris made it. Go on and find another hobby. So
Okay.
that's
Well,
kind of the thing.
it's
Because
up to him. Yeah.
you talk to 10 people, five of them are going to say yes, five of them are going to say no. That's why, as far as the Patterson Gimlin footage goes, I'm 50-50. Could Patterson have faked the suit and just got really lucky and there's nothing obvious in it that shows that it's a fake, like a zipper in the back of the suit? Maybe. Could it be an actual Sasquatch in that footage? Maybe.
Yeah.
Well, they
Not
did
a
a, they did a study of movement when it walks.
yeah.
Women will straighten their leg out. I've seen it. I've seen it on a Patterson film. I've seen it in person. A Squatch does not straighten his leg out. A Squatch will take and he'll go out so far, but he always keeps his knees bent. That's why when he walks, it looks like you put a level on his head, and his head and that level to run. They're level. So the humans, all humans that walk, consciously or unconsciously, will straighten their leg out all the way before they walk. And when we get to the point where we kick our foot up, a person don't go up as high as a squatch.
No. Yeah, you got a point there. And these people, they'll say on the phone, yes, you could,
No.
but they ought to make a suit with materials of the day and do a film and show that they can give the same muscle movements and stuff, or they ought to at least write a counter argument to every point that Munn's made.
Yeah.
It's not enough to just say yes, you could do it. You got to show me.
Yeah.
Yep.
If they would take and make us now today, I'm convinced that they can make a suit. But far as the muscle movements, it would probably take about $500,000 to make an outfit or a robot or a android that would reproduce the muscle movements.
Yeah.
Because when I had one come out in front of me a few years ago as a 6 footer, just standing there, he just stood there and you could see his muscles rippling, just standing there.
Yeah.
But when he took off, that's one thing you can say about a Squatch. They are extremely fast.
Yeah. Everybody has said that. Yeah.
Yeah,
Now,
they are.
I was gonna say Fred had another question he mentioned earlier on the chat here, the chat just between us on here. First of all, are you familiar with the Matilda
Yes.
footage? I guess.
Somewhat.
Yeah.
Yeah. I've
Yeah.
seen it on Ketchum's website.
Oh,
Yeah.
okay. Fred wanted to know what did you think about Matilda?
Yeah.
It's hard to say because the figure, I'll call it, is in a reclining position with the face turned away and so on. It's very hard to tell from those movements whether there's a costume involved or not. Um, and I'm not an expert in costumes or hair or anything like that, so I certainly can't draw a
Yeah.
conclusion.
Yeah. Now on the face, when she opened her mouth, they
Now
showed
you're
they
talking
showed
about a different one now.
Yeah, that's a different video, but I know the one you mean. Yes,
yeah,
supposedly
yeah. Yeah, it showed her with canines.
Yeah.
And every
Yeah.
Squatch I've seen that smiled at me or opened its mouth to me, I didn't see no canines.
Oh, really? Yeah.
They got human. They look like human teeth, but they're massive.
But the thing is if you look at that video, I know which one you're talking about, where it's looking off and then it looks over
Right.
the camera. If you
Yeah.
look at that, the whole face remains static through the entire video. The mouth is hanging open, the eyes don't move.
Right.
It's like the Todd Standing video.
It's a mask or
Yeah.
a bust, or I belong to some groups on Facebook of costume makers, mask makers. You can basically ask them to make. You can have a full predator suit made. Cost
Yeah.
you $1,000. But now, if you were to take that predator suit and go out into the woods and wander off, and somebody was to see you and take some pictures, they'd think it was real.
Sure. Yeah.
Anything can be made, anything can be produced. But like I said, when it comes to that Matilda video, if you watch it where it's looking off and then it looks over to the camera, the face remains static through the
Yes.
entire video.
Well, some people say that it resembles greatly a so-called Chewbacca
Exactly.
character
Yes.
from Star Wars or something.
Yeah.
That the costume isn't even original. I don't know enough about that either to say for sure, but I will say that there are a lot of people believe that.
Yeah. I have to kind of agree or lean towards that.
Yeah, because now in my area, I don't know about anybody else's area, but I got two different types. I got ones that got hair on their face, and I got a group that does not have hair on their face. Now that brings me to believe, are they the same species, or are they two different species, or is it just the genes that do something like that?
All right, good question.
Now, Dr. Haskell, now, as with you being, a skeptic, I mean, like most animals, we have different breeds or strains of, like, we have different dogs, we have different types of horses, and, all kinds of species or strains are in the horse family. I mean, yeah, I believe if being, a Squatch, I believe there's different. I mean, do you understand what I'm saying? Like, as far as could there be different strains of?
Oh yeah, yeah, absolutely.
I mean, right. I mean, like we've seen a number of sketches. I'm sorry. Go ahead. Go ahead.
No, different subspecies or just individuals may vary greatly as do humans.
Right.
Yeah, or dogs or horses even. Yeah.
Yeah. Okay.
Yeah, I've seen different sketch. Yeah.
Well, I think they adapt to what areas they happen to be in. Because I know here in Massachusetts, like say deer, we have rather small deer in New England area as compared to like say Texas or Pennsylvania. We get some pretty monster deer in that part of the country, but here in Massachusetts they tend to be a little smaller.
Right.
Well, that depends where you're hunting, because we're from Massachusetts too. 250, 300-pound deer, and where my father used to go hunting.
Well, the same in Texas even. Around where I live, they're small, but in South Texas, they're quite large. Down in the Rio Grande Valley. So yes, absolutely, there's going to be variation, and that's sort of to be expected.
Just like black bear. Black bear in my area where in my area of focus where I like to do my general research, I've seen black bear averaging around, I've seen small ones, but I've seen them average mainly around 250, 300 pounds. I mean, and from hunters, I've seen hunters take in, a good 350, almost 400-pound bears. Now, probably about a couple months ago, I seen one. I mean, I drive a Chevy Blazer, and I had one run across the road at me about 10 o'clock at night, and his back was it's the biggest black bear I ever seen alive in my area. It was well over a good foot or so above my front of my Blazer.
Wow.
And he was a big black bear. I mean, he might have been a good 500, 600 pounds, but he was huge.
They get that big.
Yeah.
You hear about the bigger ones in Alaska, but I'm sure elsewhere there are some big ones too.
Oh, yeah. Anyone else on the panel? Fred, Corey, Glenn, you guys have any questions for Dr. Haskell?
I'm not too sure, but I've learned quite a bit tonight from Doctor.
Awesome. And that's what we were hoping for, that we could shed some light on this whole situation and hopefully get people to start thinking. I mean, I know it's something I've been trying to do, but, we need somebody that has scientific knowledge that'll actually throw it out there and lay it out there. And I'm actually anxious for the paper. And
Yeah, well, I will make it known when it's out, and I'm going to buy a whole bunch of copies for people like you who've had extra interest and so on. And I'll figure out a way to get an address from you or whatever to mail it to, and then others can purchase it. It'll be available.
Awesome. That is very awesome.
Yeah, because I'd be interested myself. It's nice hearing from somebody with a scientific background who isn't biased for the existence of Bigfoot, that they keep an open mind either way. So I've enjoyed this. I really have.
So have I. It's been very informative for me too. You guys have different experiences than I do. I've never actually seen one. I've only looked for them. I mean, a couple of times, and although I'm always out in the woods looking for things, but so far nothing's popped up. So
yeah,
good work, and be careful when you collect samples. Try not to contaminate them, and we'll just try to figure out where to send them if you get anything good. But it would really be good if you had a picture with it or something.
Right, kind of showing how you're collecting it, what you're collecting, where, yeah.
Well, or preferably of the animal itself, but that's tough.
Yeah. That
Picture,
would.
either one's difficult enough.
Right. And again, I want to throw out there for our viewers. If you haven't seen After Hours with Richter, check out the show that I shared around. It's floating around. It's on Richter's YouTube channel. But check out the interview show with Dr. Haskell, Hart, and Todd Disotell. I highly recommend you watch and listen to it, both part 1 and part 2. And hopefully he broke it down to you pretty clear here. I believe it's safe to say that he spoke the truth, and it was very clear, understood. If you don't have no questions, you could try to contact him. He could explain it to you. If you didn't, I mean, I think you broke it down very clear tonight. I really think you did. I understood it, and I hope everyone else understood what you said the way I did.
Well, glad to hear that, because it is a difficult subject, and I'm happy to answer anybody's questions. I'm on Facebook. You can send me a message or whatever, and or on my blog site, which is bigfootclaims.blogspot.com. So either way, you can get up with me or comment on what I've written, and I'd appreciate comments, because it's been kind of lonely out there. I thought I'd get all kinds of criticisms and stuff, but so far nobody's pinned me down on anything, and that doesn't mean I'm 100% correct. It just means nobody's taking the time, I guess. But I don't have the last word on this. I'm still learning, and so I appreciate any constructive comments.
Now, as far as I'm concerned, if you get criticism, they're either not understanding you, or they're not understanding that they don't want to believe the truth, and yet they still want to believe what's being said blindly and what they're understanding blindly. I mean, I hope my past podcasts, I hope they're swallowing their words right now, but but what's that?
We'll see. That's very nice of you to say that, and but I'm not infallible. I can make mistakes too, so we'll see what comes of it all. When I get this paper published, I think being in writing like that and so on, that might encourage people to look a little bit deeper than they have into what I've done.
Right. And again, like I said, I'm going to say it one more time. Check out After Hours show. It was from 2014 with Todd Disotell and Dr. Haskell Hart. They broke it down. They threw it out there, and when you guys hear Dr. Todd Disotell talk about Bigfoot 101, well, it's what Richter called it, because I started stated on the bottom as Todd was breaking it down about Bigfoot researchers. I totally agree with what he had to say about that. And then, of course, what's her? Natalie Reagan, was that her name who popped on there?
Natalia, yes.
Natalia, yeah, okay,
They
Natalia
were on
Reagan.
the $10
Yeah,
million Bigfoot show.
Yeah.
right, and yeah, she was she's a primatologist, and I mean, she was pretty fair on her words, and she spoke, she gave her 2 cents about Bigfoot researchers and how to better conduct being a researcher in general, as far as research subjects, that you're affiliating your research to, and I mean, they couldn't have said it any better. So, I mean, definitely check that out.
They did a good service by educating people on how to collect samples and do field work.
Absolutely, absolutely. I think we could all learn from that. I mean, I know I did. I mean, I take everything I can and learn from it. I mean, it's awesome, and I hope our viewers did too. I mean, man, I almost don't want to end the show. It's been so great, but I want to thank you again for agreeing to come on tonight. It was awesome to have you. Maybe another future show I could get both you and Todd in here, and we could throw it at them again.
Yeah, well, it was my privilege to be on the show. I enjoyed it too. I learned some things too, so it's a good deal all the way around.
Yeah.
Absolutely.
Yeah. Hey, stick around for about 5 minutes after the show. I got something I want Daniel to send you.
Oh, no. Okay. All right. Well, again, viewers, thank you very much. If you haven't done so, subscribe to ECBRO 98 here on YouTube. And again, check out After Richter, the After Richter show with Todd Haskell and Dr. Haskell Hart. It's a show you don't want to miss. It's from 2014, and I hope you guys enjoyed the show and learned something from it tonight. Everyone have a good night, and God bless.